other types of things that you people look at. And so in this pack, I'm going to be basically talking about a lot of functional genomics. And I use the term multi-omics because there's many different types, as we'll talk about, ChIP-seq, RNA-seq, and so forth. So first of all, I thought I'd just give you an overview of like what annotating the genome is, because a lot of these sort of techniques are really about annotating the genome. They're things that we get readouts from different parts of the genome, and we use it to kind of annotate. And so I thought I'd just kind of start off with a little idea on that. So what do we mean by the word annotation? I think it's kind of nice to think about it in terms of how we would annotate a text. So here, actually, I have an original text. This is Watson and Crick's text from 1953. And in this particular text, they're talking about the structure of DNA. And then over here, I have the text on the initial sequencing analysis of the human genome. And how might someone just annotate these texts? Well, they might look for the repeated words, in here. They might look for, they might sort of look at just the kind of functionally annotate different parts of the things. This is obviously the title of the two things. This is an author list. This is kind of the introduction and so forth. So you imagine if you had a highlighter, you might be coloring things differently depending on the function and so forth. And then you could imagine also comparing these texts, and you of course would see the name of the journal being the same. You would see maybe the DNA double helix being the same, but not exactly the same things. And these are simple things you might do if you were functionally annotating things or annotating these texts. And so now let me talk a little bit about how you might think about this in terms of annotating the genome. So it's a sort of same idea. So for the genome, we have 3 billion letters, and there's sort of two basic techniques we have for annotating it. One is we have functional genomics, which we're going to talk a lot about here. And here you get some readout on the genome. It's almost like a highlighter pen sort of saying, well, there's a lot of, say, binding happening here, or there's a lot of transcription happening here. And in bioinformatics, what we do is we get a very noisy readout. We're essentially doing signal processing to process this into a smoother signal. And then we kind of, to some degree, threshold it to get like little blocks of annotation. This is where there's a lot of binding. This is where there's a lot of transcription. And then we might group those together. On a high level, that's kind of one of the things we're doing. The other thing we tend to do is to take the genome and compare it with, well, we can compare it with itself. We can compare it to similar genomes within, say, the human population, or we can compare it to genomes in other organisms. And then what we basically get in any of these comparisons is we get regions that are similar. And so if we obviously compare it with itself, we'll get kind of repeated blocks within our own genome. If we compare it between two people, we'll get conserved blocks within the human population. And then if we compare it between different organisms, we'll get conserved blocks that are preserved over longer time periods. And now the way these figures are drawn, it looks very kind of easy to do the comparison. It's a little more complicated. I mean, when you're comparing a very distant organisms, it's a more complicated comparison. It's a very different type of thing than when you're, say, comparing within the human population. But on a high level, that's kind of what we're doing. Okay, so now let's talk about that kind of marker type of thing, kind of how we're getting a readout on the genomes. First, let's talk about RNA-seq. And I'm just going to give you, I'm not going to go deep into these techniques here. I'm just going to touch a little bit into them just to give you a sense of what happens in these techniques and what type of processing we tend to do. So as you know, in RNA-seq, we're getting a readout of the transcription, and we map it back to the genome. And the result is we get figures that look like this, where you'll get sort of a lot of reads piling up at certain spots. Usually these are in exons or places where there are genes. And what we often do is we aggregate all the reads on a particular exome or on a particular genomic annotation, and we'll get something like an exon expression level or a gene expression level. And usually sometimes they describe this in RPKMS and some sort of normalized in relation to the sequencing coverage and so forth. And so this is a typical type of thing that people will do. Another thing people will often do is they'll look at change, say in a person between two conditions, or they'll look at two different individuals and so forth. And these are typical types of things where they'll look at the log fold change, but they might want to look at it relative to the amount of underlying expression that you have. Obviously, if you have more expression, you have more confidence in the change. Very typical. There's a lot of ways of doing this. I just list some of the popular approaches here. It's more of just to kind of complete things. And I have some references here. DE-seq is a popular method. It's based on the negative binomial distribution. These use different statistical models, EDGE-R, and so forth. So I just want to give you a little bit of a sense of these methods. Now I'm just going to go on to ChIP-seq. So ChIP-seq is a little bit different. The processing we're doing, because we don't have as clear a sense of, we know these are genes, we know they're not. So in ChIP-seq, we also get a signal on the genome, but it's usually less concentrated in particular annotations and more of something kind of spread over the genome. Okay, so it looks more like this. And what we're often trying to do in ChIP-seq is to take this kind of wavy signal over the genome and really turn it into some form of annotation block. Or people use the term peak calling. So here's a picture of what tends to happen. You have this very noisy signal, and at a simple level, you could imagine just thresholding the signal, and just all the pileups above a certain height. People usually do somewhat more complicated things. I mean, they do. They usually they might have a kind of control that they might imagine is like the background in the genome, and they sort of think of that control as giving rise to a certain amount of reads kind of randomly. And then they compare the number they observe relative to the control. You can imagine some sort of like a binomial type of test or some sort of statistical test like this. And they often will do something like averaging over thresholds and so forth. And again, I'm giving you a very simple view of this. I just want to give you a sense of the type of calculations people do. Now, a big thing that people do in ChIP-seq, which is very important, is they often look at transcription factors that bind in the genome. Okay, so they're looking at now the binding of, say, a transcription factor. And what will happen here is they'll find a peak, and so they'll find different peaks for the different transcription factors. Okay. And then what they can do, that's from, say, looking at raw data that looks like this. And then what they can actually do is they can take these peaks and they can start to build networks. And we're going to be talking later about networks, but it's useful to think about where the networks come from. So imagine you take a transcription factor and you find all the peaks. You might get something that looks like this. Well, if the peaks are right upstream of a gene, say in a promoter, you might say this transcription factor operates through that promoter and regulates that target gene. Okay, so you could create an edge from that TF to that gene, and therefore you could build a network. Now, one of the key things to realize is that for transcription factors and regulatory networks, they can regulate other factors, so that you'll often have one factor regulating another factor and so forth, building a complicated network. And this is a picture showing a view of the human regulatory network, which had, at the time this was made, I think about 500,000 edges. Now, these are for what's called proximal edges. You often have regulation, though, in the human genome, particularly at a distance from a gene. The transcription factor can bind far away, and usually these sites tend to be called enhancers and so forth, and they can be very far from genes. And I think very importantly, it's not so obvious what gene they go to. So there's a very complicated discussion of pairing up what's called distal elements with promoters or with genes. But if you do that, you can then create also a network where a transcription factor regulates a gene through an enhancer, and that creates a network of distal interactions. Now, one of the things that is very important in looking at these ChIP-seq experiments is to try to understand what's signal and what's noise. Now, since we don't know where there's genes, we don't really know ahead of time where there's peaks, it's very hard to get a sense of, oh, well, that little bit of signal there is noise or it's a real binding. And so people have spent a lot of time thinking about replicates and doing an experiment more than once, and then maybe looking at concordance between replicates. And they've introduced this notion of irreproducible discovery rate, which is basically like as you go down the rank list of, say, peaks or features in a ChIP experiment, how reproducible they are. So if you rank all the ChIP, the peaks in terms of their strength, and you say, oh, the top ranked ones, they agree, and obviously at the bottom ranked ones, they probably don't agree because you're more into the noise. You can kind of look at the fraction of things that aren't agreeing as you're going down that list. And that creates this IDR graph. And you can see at some point when you move from kind of real signal to noise, if it was noise, you would only expect a small fraction of agreement. You would maybe see a jump in this IDR. And that's the idea behind it. Another thing people have spent time doing with ChIP is really trying to think about how to call features and parse these signals at different levels. So for instance, here's a signal here. This is for an actual histone marker feature. And you can see if you just look at the signal, it's not so obvious, well, what is a peak? Is this whole thing a peak, or is that spike a peak, and so forth? And so you can smooth the signal. Obviously, you could smooth it in bins of different width. And as you smooth the signal, you're going to get different features. When the signal is very smooth, this entire region, say, will collapse into one big peak. When it's not so smooth, it will be maybe 3 regions and so forth. And that's an important idea, and people have different thoughts on that. This is just a nice picture showing how the window smoothing kind of looks. If you look at it, you can kind of smooth with a greater window, and you can see that you can get features at high resolution, and they disappear as you smooth, and they turn into very broad features. And that this is a characteristic of ChIP-seq analysis. Now, another thing people want to do with ChIP-seq is they like to do some integration. So for genes, it's usually pretty much the RNA-seq we're looking at, but for looking at the binding sites in the non-coding genome, there's many different assays. Now a lot of this integration is going to be in the framework of various machine learning approaches that we're going to talk about later. I just want to give you a little bit of a sense of what people do here, just to give you a little bit of a sense. So the idea, for instance, let's say you're looking at finding enhancers. Often there's many different types of readouts you'll get for the enhancer. You'll have the different biochemical annotations. You'll have conservation, what we talked about. We haven't talked about this in detail, but there's many other types of information people might do. For instance, there's new technologies in genomics now that let you get very targeted readouts from sort of reporter assays and so forth. So often, a lot of the times you'll find, say, an element from one technique, it won't agree with another, or you'll want it to integrate a lot of different techniques. It's very much like this kind of looking at an elephant from different, different perspectives. One thing's looking at the trunk, and another's looking at the foot, and so forth. There's many different technologies for doing this. I wouldn't even think to go through all of them in this class. I just list a lot of the different methods. There's a very well-known sort of method of looking at a lot of chromatin signals called chrome-HMM, and I think the key thing to realize here, it uses an HMM, or hidden Markov model, just like the one we talked about earlier for looking at sequence patterns or looking at structural variants. You can also look at histone marks and so forth. And so and as we'll talk about later, some of these are supervised, some of these are unsupervised, and so forth. I just want to give you a little bit of a sense of the sort of simple ways of doing it. The ENCODE project is a big project that tries to annotate the human genome. it has a very simple-minded way of calling a lot of these features that just basically looks at overlaps. So there's some, you look at it at an element in the genome, and you can just look, oh, is there a signal in this histone mark, and also CTCF, yes or no, or maybe there's only signal from one histone mark and so forth. And so so just on a very simple level, you can imagine just overlapping things, which is a very reasonable thing to do. And then there's somewhat more sophisticated ways of maybe you could also look at the signal shape. And so this particular method, which is called match filter method, it looks tries to fit a particular signal shape profile. Because, for instance, one of the best known histone marks is K27 acetylation. It's known to have a kind of characteristic double peak structure. And so you can imagine taking a profile for that type of shape and just kind of moving it across the genome. So this particular method uses that type of approach. Okay, so now I'm going to tell you about some other types of things people do. Now, so far, all of the calculations that we've talked about in functional genomics have not looked at variants, right? So they're just looking at the signal. And actually, the underlying sequence, the underlying genomic sequence is not even there. It's almost just an afterthought that the sequencing machine is getting a readout on that. The calculations we've talked about are just, they're really not looking at variants. But there is a set of calculations that people do where they try to relate the variant in the person's DNA to the functional genomic signal. And the simplest one is called allele-specific binding expression. And the basic idea here is, for instance, if you have two copies of the genome, you have your maternal copy and your paternal copy, you could imagine, for instance, a situation where a transcription factor might bind to one of the copies, but not the other, because there's a variant. And likewise, you could imagine a situation where, say, here's a gene, and you have a promoter upstream of the gene, and maybe one of the copies, say the maternal copy here, there's a variant, so that turns the gene off, and in the paternal, you have the gene on. Now, you might say, well, how can you determine this? Well, it turns out it's really easy to do that with next-gen sequencing, because if you look at a chip experiment, what happens in this chip experiment is you get reads, and each read relates to the binding of that transcription factor. Now, actually, these reads actually contain the sequence from which for the different diploid copies of the genome. So, for instance, if they come from the maternal copy, they would have one letter, and if they come from the paternal and there was a difference, they would have a different letter. So when you make your read stack, you'll actually see something that looks like this, where you'll have the maternal and paternal copies. And then you can simply separate out the signal for each of those types of things. And that will give you what's called your allele-specific binding or expression information. Now, these calculations, of course, when you drill into them, they get somewhat complicated. I just want to introduce one small notion. So the notion is that imagine you have a gene or a site in the genome that you have a heterozygous SNP in it, but the heterozygous SNP doesn't make any difference. So there's a binding site, there's a SNP, but the transcription factor doesn't really care. So then you expect to have an equal number of SNPs. And so the expectation is if you sequence to a depth of, say 10, you get 5 from the maternal, 5 from the paternal, right? But it doesn't, of course, work like that. It's really statistical. So you get, on average, 5 and 5, but sometimes it can be 4 and 6 and so forth. And so there's usually what's thought of as a binomial expectation, okay, in the background. And then if you actually had a true shift in allelic behavior, you would see it at the tails here. So for instance, if you saw this peak over here, where you have a lot of things that have a very high expression for one allele, but don't have expression for other, that's a true allele-specific, say, expression or allele-specific binding type of situation. Now, it gets even more complicated because and this is important to know, when you map the reads to the genome, remember you're looking for how the reads map to the maternal genome and the paternal genome. But imagine you're mapping them just to the reference genome, okay? There's a bias because if the reference genome contains the, say, maternal allele or the paternal allele in it, you will preferentially map to that. It's easier to map to that, and therefore you'll seem to get higher expression or binding for the allele in the reference genome. And that in this type of plot, that will lead to a kind of imbalance where you'll seem to get, say, more overall allele-specific expression for, say, the paternal allele than the maternal allele. And that's not real because you wouldn't expect that type of situation at all. You expect it to be kind of even. And that has to do with what's called reference bias. And, it's a complicated topic of how people deal with reference bias. One of the simplest ways of doing it is actually to build custom genomes up for the genome that you're actually doing the functional genomics on. And in particular, you would actually build a diploid genome up. So that actually has the maternal and the paternal copy of that genome. This is hard to do. And then you would map the reads back to those, to that diploid genome. And then you would no longer have a bias because you have the perfect copy of the maternal genome, perfect copy of the paternal genome, and then the read would map where it perhaps should. Now, it sounds all very easy. It's actually somewhat hard to do this. But in any case, it's kind of conceptually the right thing to do. So let's talk a little bit more about other calculations you can do with variants. Now, allelic calculations, you just need the functional genomics, that's RNA-seq or ChIP-seq, from an individual, and you're comparing essentially the alleles within an individual. Another calculation you can do is called an eQTL, or say CQTL, a QTL calculation, where you're looking at a cohort, okay, that has variants in them, and you're looking overall in the cohort, how are the variants at a position related to, say, the gene expression or binding level, okay? And so, for instance, if you have a SNP at a particular position, you can look at all the people in the cohort and say, oh, is in a particular person, is that location, say, ref, or is it alt, or is it heterozygous alt-ref? And then you can see in the cohort what's the, say, level of gene expression for those people, and if you see there's a correlation, you can just basically fit a slope to this line, and that slope becomes an eQTL. Now, there's more complicated things. People can, of course, correlate any SNP in the position with any gene's expression, and it gives rise to a huge amount of testing, and there's a big issue with multiple testing. But it's useful to think about kind of what you might call cis-eQTLs that are located right upstream of the gene in the promoter where you might expect them, and trans-eQTLs, which might reflect a much more complicated relationship between that SNP, which is far away from the gene, and how it's affecting the gene expression levels. And this picture shows that the QTL calculation in a little bit of a different way in terms of a distribution for each thing. Oh, okay. So now I'm going to stop